Thanks for the organizer for inviting me. I'll be talking about structural variants from the perspective of a clinical geneticist. And this picture here is totally unrelated to what I'm going to talk about. But I show it to try to, it's a complex pitch to try to attract students to come work in my lab. So what I tell them is that, you know, in the winter, Montreal is super cool. Like, you can ride your bike on a frozen lake. And then late spring, so this was taken last week, you can still ride your bike on the same frozen lake. And believe it or not, in the summer, you can swim in those lakes. The water does get really warm, but it's, you know, it's totally crazy and schizophrenic place. I don't even know why people have ever colonized that area of North America. So the outline of the talk is why are CNVs overrepresented in the patients that we see in the clinic? And it seems like a really naive question, but you'll see that it's actually a very complex issue, and except for the amazing work that Anne did. But, you know, it's like a lifetime investment for the vast majority of the variants that we see in the clinic. We do not know why they are present in our patients. So what are the effects that these mutations are driving and that are then bringing the patients to be referred to the clinic? So I'm just going to go really quick over this because we've had an amazing introduction, but just a very brief reminder, structural variants compared to any of the other molecular mutations are super easy because deletions decrease expression and duplications increase expression. And when you're looking at SNVs or SNPs, you never know exactly what they're doing. So I think that's the really important part about structural variants, because the molecular impact is very straightforward. This is a list of your usual suspects that are formally associated with schizophrenia. I could show you a smaller list with autism. And yes, they are highly overrepresented. So statistically, it is absolutely clear that they are more frequent in patients than they are in the general population. But why? Except for a few CNVs, we don't really know. So, you know, can we try to understand what are the effects of these mutations on cognition and behavior? Yes. So I think the presentation by Anne and the presentation by George were amazing examples of what you can do. I'll just give you some similar results from our work on one of the other really common CNVs, which is 16p11.2. And what you can see here, this is old work already from 2011, but if you take very simple traits that you can measure, such as BMI or brain volume, if you look at the intrafamilial controls and you look at deletions or duplications, you really have this sort of quantitative gene dosage effect. So if you decrease expression, you're increasing your BMI, and if you increase expression, you're decreasing your BMI. And it's a very quantitative relationship. And same thing for brain volume. If you decrease your expression of that region, you're increasing your brain volume, and if you increase expression, you're decreasing it. So you can do this with multiple traits, and you can quantify exactly the effect size. It's about 0.8 standard deviation, so it's a large effect size, and it's clinically relevant. You can also do this with cognition. This is some earlier work, but it's been replicated. It's a very stable effect. It's 24 points of IQ. That's what a deletion does, and it doesn't really interact with anything. So what you can see is that this is the distribution in the general population, and this is the distribution in 16p. It's just sort of a simple shift. So you subtract to where you were without the deletion, and then you take 24 points out. you can look in more details at different types of cognitive traits to try to understand, you know, okay, IQ is a really important element for clinical referral, but maybe there's other things. And so what you find is, yes, phonology is very sensitive to the 16p11.2 region, so you really lose 1.5 standard deviations in phonological skills. But on the other hand, you can see that, for example, duplications improve short-term and long-term verbal memory. So that's probably not why they're referred to the clinic. You can also see that duplications, this is corrected for IQ, has absolutely no impact on language. But every kid with a duplication referred to the clinic has language delay. So that really tells you that to be referred to the clinic, you need a multidimensional set of disabilities, but the CNV is not doing everything. It's contributing to some of the problems of the child, but not to all the difficulties of the children. And I think this is a very important point because you hear all the time, well, the genetic mutations, they have this really broad impact. Yes, they do, but they're a lot more specific in terms of cognitive and behavioral effects than what we think. It's the clinical referral which is driving this. So to be referred to the clinic, you have to have multiple disabilities across several dimensions. If you're just dyslexic, you're not going to be referred to the clinic. You can have a job, you can even be a professor, but if you're dyslexic with a little bit of cognitive deficits, a little bit of anxiety, and then who knows something else, then you stop functioning properly. Okay. The brain structure studies that have been conducted, these are two studies that we conducted in 2015, 2018. What they show is, again, a very robust effect of this, of gene dosage at this region in brain regions that are previously well known in psychiatric conditions. So what you see here, for example, the insula, the transverse temporal gyrus, the effect size is about 1.5 standard deviations. So very similar to the effects that you see on cognition and behavior. You see the same quantitative effects at the structural level in the brain. And these brain regions are well known by psychiatrists. Actually, this region here, which is modulated by 16p, is also the top region that is affected across all psychiatric conditions. So if you pull everything together, schizophrenia, bipolar, major depression, and substance abuse, those are the regions that you're going to see pop up. So that might be also a key to why some of these structural variants are putting you at risk for so many different psychiatric conditions. Using these mutations also allows you to sort of infer longitudinal effects without having access to longitudinal data. So the advantage of some of these mutations is that you sample, you ascertain them in children, in adolescents, in adults, and then in older people. And you can, if you have a good way to normalize your data depending on age, you can see when some of the symptoms start appearing. And so this is the brain imaging data that you saw before, but then normalized on the National Institute of Health developmental data, which is a structural neuroimaging data set, which starts at the age of 4 and that goes all the way to the age of 23. And what you see is that some of the top regions that I've been showing, for example, the insula here, this is starting at the age of 4 all the way to 24. These alterations, so this is the control population, these are the deletions, and these are the duplications. You can see that the alterations in these regions, they're parallel and they do not change with age. So at least what we see is that at the age of 4, these alterations are already there and they don't evolve between 4 and 24, despite the fact that there's many different symptoms that start appearing during adolescence and then early adulthood. And you can see this for all the structural rearrangements, the brain structural rearrangements that we've looked at, it's all parallel. So I'm not saying that there's not going to be any age effects, but so far we haven't been able to find any. And so now we're really interested in scanning babies, newborns, to see if some of these top schizophrenia alterations that have been very well characterized in adults are already present in these neonates who are at very high risk of developing late onset psychiatric conditions. You can also explore more general impacts of CNVs on brain structure. So here you have three regions, the 1Q21.1, the 16P11.2, and then 22Q11.2, which Anne has talked about. And what we're representing here is the distribution of regional brain alterations impacted by deletions on the bottom and duplications on the top. And what you can see is that for each of these genomic regions, the deletion has about a 1.7 to 2-fold increase in the alteration of brain structure across the brain for all of these genomic regions. So there seems to be sort of a general principle, at least at the brain structure level, where when you apply gene dosage, you're distorting the brain with a very fixed ratio, you know, a 2-to-1 almost ratio between duplications and deletions. And I think this is sort of where our hypothesis is that you can see this across the genome. So anything you delete or duplicate, you're going to have this distortion of the relationship between regional brain structures. You can go further and start looking at functional brain connectivity, and I'm just going to gloss over this a ton of work that Clara has been doing for the past 2 years, and there's a poster if you want to dive into the details of this. But here what we did is we looked at 16P and 22Q resting state functional MRI data, and we also looked at 3 psychiatric conditions: autism, ADHD, and schizophrenia. And these are sort of complicated plots, but what they're showing are the effect on 2,080 functional connections in the brain, the effect of the deletion 16P and the duplication 16P. And what you see here is that globally, all these connections are increased in their connectivity with the deletion, and they are decreased in their connectivity with the duplication. So you have also sort of a mirror effect at the functional level, which is very robust and almost has the same effect size as what we see in the structural alterations. And if you look at the 22Q, it's also somewhat of the same effect, except that it's completely flipped around, and the deletion is decreasing overall connectivity while the duplication is increasing it. And if you look at the connectivity patterns of these 2 deletions, the 16P11.2 and the 22Q11.2, the connectivity patterns are not necessarily specific to these regions. And if you start comparing them to schizophrenia and autism, what you see is that individuals with schizophrenia and autism have an increased similarity to the connectivity alterations that you find in 16P and 22Q. And if you compare all of the connections of individuals with schizophrenia, autism, and 16P and 22Q, you end up with this complex Venn diagram of patterns where you have for each Venn the increased similarity. For example, this Venn here shows an increased similarity between individuals with schizophrenia and the 16P11.2 pattern. And here are listed all the regions that are driving this similarity. So I'm just showing this because as we gather more data across the genome on these CNVs, and we have a better understanding of what are the clear patterns for the different psychiatric conditions, we're going to start being able to understand what are the basic building blocks that are necessary to drive disabilities in our patients. And you know, I keep talking about effect size, and I'm pretty obsessed about effect size, because in the clinic, I think that as you in this room are going to start prescribing, you know, genetic tests in the clinic and start doing genetic counseling, I think we need to start moving away from this pathogenic, non-pathogenic, into sort of a quantitative genetic counseling to our patients. And this is a good example. For example, this is the 15q11.2 deletion, which is a very common CNV which is clearly associated with schizophrenia, with neurodevelopmental disorders. And if you just use here the case series reports on this deletion, where what you're going to give to the patient is, well, it's a pathogenic CNV with incomplete penetrance, and you have a 12% risk of autism, a 42% of intellectual disabilities, et cetera, et cetera. If you really use the true quantitative data that has been gathered for this CNV, you see, in fact, that the risk for autism is 2.4%, the risk for ID is 3%, and the risk for epilepsy is 2%. So it's a very different message that you're giving to the family if you're using the right quantitative data. And we now don't even, we report it back without really reporting it back. And this is a paper that is in press where we suggest that this should be reported back quantitatively, saying, you know, it's yes, it contributes, but such a small proportion to the neurodevelopmental symptoms in your patient that it's not really worth using alone. So I'm just going to skip this. Yes, in the future, when we have algorithms adding up all these different genomic variations, it'll be really relevant to add the 15q to this algorithm, because it'll be part of a larger spectrum of mutations, and we'll have some better predictive risk with algorithms. Okay, so let's see. It's 12:30. So that was the first part of my talk, so I'm a clinical geneticist, and I report CNVs back and mutations back to patients, not every day because I need to do research, but almost every week. And I've spent a couple years working on 16p, and Bassett, you know, dedicated her life to 22q, and it's great, and it provides incredible insight. And I think, you know, these CNVs have really helped us understand, you know, how genomic variation can really affect cognition, behavior, and brain functioning. But there are thousands and millions of CNVs out there, and when we're going to start implementing a whole genome, we're going to be drowning in mutations. And for the vast majority of mutations, there's almost no data, and you cannot really do any proper genetic counseling. So, you know, I'm 47 now, and I have like 20 more years of research activity. I spent already 3 or 4 on 16p. It's just, you know, you do the calculation in the morning, it's just not going to work out. So we need other strategies to get insight on all of the genetic variation and how they impact cognition, behavior, and risk for psychiatric and neurodevelopmental disorders. This is just an illustration here of the problem that we're facing in the clinic. So in the hospital where I am, in the pediatric hospital, we do about 4,000 CGH arrays per year. So this is roughly 3 years of work. And these are the genes, the unique genes covered by deletions. There were only, I'm not counting the ones that are covered twice. After 3 years, these are the ones that we've actually reported back to families saying, oh, this CNV is pathogenic. You've covered almost 8,000 unique genes that are deleted in one center. That's like a third of the genome. It's a huge proportion. And the good data, good quantitative data on cognition, behavior, and risk for disease on these 8,000 genes, it's a minute fraction. So we're facing a huge problem, which is the discordance between the amount of genetic testing that we're doing and the actual good information that we have. So we've been, toiling with, what can we do? How can we do this? I think the UK Biobank is a great resource. You can start looking at CNVs. And just so you understand, this is a GWAS. And when you're running GWAS, each individual SNP is frequent enough to allow an individual association study. So you can run a linear model for each SNP, SNP by SNP, okay? And so you get a p-value for each SNP. For CNVs or rare variants that we're going to get on the whole genome, you're not going to be able to do that because each mutation is so rare that unless you genotype entire China, you're not going to get enough observations to run a linear model. So maybe the next generation will have access to the whole genome of the entire planet, and you'll be able to do that, but we're not going to be able to do that. So we need other strategies, and the ones that we've been tackling in the lab is say, okay, and this will be a recurrent theme in the next part of my talk, is that the specific effect of all these millions of mutations, we're never going to know them. I'm not going to know them in my lifetime unless there's some quantum leap in technology. But maybe what we can get insight on is the effect of global genomic dimensions that we can score using different types of annotations. And these are the annotations that we've started to use in the lab to try to understand how does the genome as a whole impact cognition, behavior, and risk for disease. So you have these fitness scores, and you can ask more questions on these, the PLI, RVIS-DEL score. These are, especially the PLI and the observed over expected, these are updated fitness scores that are very powerful. You have a whole lot of new brain expression scores. There's all the new scores from the Psych ENCODE also that are coming out, and there are old categories also of gene function scores. And the initial study that we did in collaboration with many people, including Thomas here, who only goes to the lab with his Academy of Science coat on. But anyway, so this is the first study that we did using Imogen and Saguenay, which had the advantage of being two cohorts with very good cognitive data. And the question we asked is, can we develop a model that explains and estimates the effect of any CNV on general intelligence and IQ? And we ran multiple different types of models, and the one that seemed to be the best in this was a model using a fitness score. And we can go into more detail of this, but everybody gets a score based on the number of genes deleted or duplicated and the fitness score of each of these genes. And what it tells you is that if you delete anything in the genome, and if that anything amounts to about 1 point of PLI, you lose 2.7 points of IQ. So it doesn't seem like a lot, but you'll see that it grows very, very quickly. What it also tells you is about a third of the genome impacts IQ by at least 1 point. And we can discuss this later, but there's new theories of the effect of the genome on general intelligence and other complex traits. And if you challenge this model and you say, okay, I'm taking out all the recurrent CNVs in the training test, and I'm going to see what the model tells me about CNVs that it's never seen before, but that some clinicians have gathered empirical data on. You get this concordance. So this is what the model estimates, and this is what has been reported in the literature. And what you see is that there's actually a really good concordance. Here you have the 16p, the 22q. We spent a lot of time trying to characterize the impact of 16p, showing that it takes out 24 points of IQ, and the model's never seen a 16p in his life, and it tells you that CNV takes out 24 points. It also tells you that the 22q11.2 takes out exactly, I can't remember, 30 points or something like that, which is exactly what it does. So by observing the impact on IQ of a variety of different CNVs in the general population, this model can predict and estimate very accurately what pathogenic CNVs do in the clinic. This is unpublished now, but what we've been doing now is trying to replicate these initial findings. And the first thing we did is like, okay, so how does this model fare in disease cohorts? So let's take an autism cohort and see, if it works exactly the same way. And what you see, this is like a mini meta-analysis using 4 autism cohorts. The impact, so this is the mean impact of every point of PLI, and the impact is exactly the same in an autism cohort. So the general effect of deleting 1 point of PLI anywhere in the genome is exactly the same whether you're from the general population or from an autism cohort. And we haven't done it in schizophrenia or other cohorts, but our guess is that it's going to be exactly the same. I'm just going to jump this part and go directly here. This is, again, running this model now on a much larger data set of about 23,000 individuals across multiple cohorts. And what you can see also is that basically as soon as you have a cohort that's powerful enough, you end up with 0.18 z-scores is exactly 2.7 points of IQ. So again, there's this sort of very intriguing effect of a genome-wide principle of deletion and the effect of haploinsufficiency on general intelligence. And everywhere we look so far, we haven't been able to show a different effect, and the effect size is extremely stable. Now, the duplication, we didn't have a power in the initial publication to look at the effect of the duplication, but now that we have enough power, what we're seeing is that basically, first of all, it's the same haploinsufficiency scores that best predict the effect of any duplication on general intelligence. And the effect size is about 2.5 times smaller. So any point of PLI in the genome that you duplicate, you decrease your IQ by, we're thinking 0.9. Maybe with the UK Biobank, we're going to run this at a much larger scale. It might be 1 or 0.95, but it's a very accurate effect. And I'll show you some concordance analysis on this, especially with trisomy 21. There's something very intriguing about it. So then we're like, okay, so yeah, that's the general population. As George highlighted, a lot of the general population cohorts, or even the autism cohorts, they're biased. Some of the most severe cases are not in there. What happens if we take, the hardcore neurodev patients from the clinic and we run these models? So this is what we do. We have a running study which is called the CNV family study, where we recruit any large CNV in the clinic, and we recruit all family members who carry the CNV or family members who do not carry the CNV and who act as intrafamilial controls to sort of adjust for all the additional genetic background and socioeconomic background present in the family. We do this because in the clinic, as I said, people who are referred to the clinic are referred because they have multiple disabilities, and it's a little bit of IQ, a little bit of social cognition, a little bit of anxiety. You need that multi-level impairment to be referred to the clinic, and we think that in fact most mutations can't do that alone. So. That's why we're running this family study to understand, how rare variants in combination with other variants are generating these complex neurodevelopmental symptoms. And when you look at some of these studies, this is the Danish study looking at the comorbidity of other psychiatric conditions in first-degree relatives with people who are diagnosed with autism. So for example, this is the cases you see. These are the odd ratios. So if you have a sibling diagnosed with autism, the probability that you have any other neuropsychiatric condition is increased by 2.6. And you can look at all of these conditions. So everything's sort of increased in first-degree relatives of individuals who have autism. And in the past, one of the main interpretations of this is that, the genetic burden, which is associated with autism risk, is an aspecific risk for psychiatric conditions. So it puts the first-degree relatives at risk for many other conditions. But in fact, there's a huge issue in psychiatry, which is a sort of a mating which complicates our interpretation of the association between mutations and psychiatric conditions. So this is a fascinating study that you should definitely read if you haven't read it. And what it shows is that if you're a male proband and you have a diagnosis of ADHD, the probability that your spouse has a diagnosis of ADHD is 45%. If you're a male proband with schizophrenia, the probability that your spouse has a diagnosis of schizophrenia is also 44%. And for ADHD, it's going to be 21%. Autism, 42%. So when you're running these family studies and you see these kids walk into the clinic, yes, you find a mutation in this individual which is maybe inherited by one of the two parents, but that mutation is attracting other mutations in the spouse. So there's going to be a lot of mutations in the siblings and in your patients that are completely unrelated to the symptoms that you're looking at. And assortative mating is probably the #1 confounding effect in the genetic studies, in the interpretation of how we link the mutations that we find in our patients to the symptoms that are present in the patients. So when we went back to our family study and said, okay, we're finding CNVs in our patients, and our patients are a lot more severe. When you correct for the genetic background in the family, what you find here, so this is the centrosine core, it's not completely updated, but the effect of every point of PLI deleted is even a little bit lower than what you find in the general population. So the take-home message of this is that we are in general overestimating the impact of the single mutations that we're finding in our patients, because in the clinic, most of these mutations are hitchhiking in a car that is full of psychiatric risk factors. And this hitchhiking business is part of the disease. It's not some sort of incidental phenomenon. That's what psychiatry is. It's really, aggregates of risk factors that are attracting each other for all sorts of crazy mechanisms. But anyway, so far, we're still at the same effect size for the effect of haploinsufficiency. So this is, some very recent analyses that came out like a week ago. But what it shows is that if you take the mega-analysis of about 23,000 individuals, and you look at, the average effect of each point of PLI that is deleted or duplicated on IQ, and then you say, okay, these linear models, yes, they're driven mostly by large CNVs. What happens if you exclude all the individuals in the cohort that have a CNV that scores 10 points, 8 points? For example, 22q is 10 points of PLI. 16p is 8 points. You go down, you exclude more and more and more and more. What you find is that if you exclude, the impact of each point of PLI remains exactly the same. And at the end, if you exclude everyone except those who only have 1 point deleted, you still have this exact same effect. So it doesn't matter how many points you delete. It's this sort of monotonous, linear, incremental effect. And I think that's what's really troubling for molecular biologists, because you know, when you're doing molecular biology, what you see everywhere is interactions. You see interactions everywhere. And what we see is, we never see an interaction. We always see additive, additive, additive. So, and this is something in the field, there's something, conceptually this is a big issue in the field. So each time you do population genetics, everything you see is additive. Each time you do, you know, a cell genetics study, what you see is, you seem to see real interactions. And so there's ways to reconcile these observations, but probably what's going on is that everything is interacting with everything. In the end, there's so many interactions that it just becomes additive. It's not a very good explanation, but there's a lot of interesting mathematical investigations of this. Now, if you go back to the duplications and you start running concordance analysis, I think this is getting now very interesting. So the first thing that you realize is that in the literature, there's almost no data on the impact of duplications on cognition. I mean, in 2019, we have no data. There's a handful of them, okay? So we did still try to gather as much data as we could, and a lot of these points are mostly single case reports, okay? So what you see is that this is what the model predicts, this is what you report in literature, and what you see is almost everything is overestimated in the literature. So in the literature, they say, whoa, you know, this guy came in with mental retardation, he's got a really big duplication. Our model says, well, you know, your duplication doesn't even take out 10 points of IQ. So we're like, ooh, who's right, who's wrong? So I think this is open for discussion. So we really think that we're right. But this is why we think we're right. The one duplication that has been really well studied for half a century is Down syndrome. And Down syndrome is right here. So the model has never seen Down syndrome in its life. It's actually calculated based on 1 point of PLI. Down syndrome is 50 points of PLI, okay? And Down syndrome is about 55 points of IQ. So it's exactly what the model predicts. And if you go to the chromosome 21, there is opportunities for interaction. Okay, there's 260 genes. There's got to be interactions. 34 megabase plus 10 megabase of a short arm on which there's probably not much, but there could be all sorts of interesting regulation stuff. And we've been looking for the Down syndrome critical regions for half a century, and you know, we found it, we didn't find it, maybe it's there, maybe it's here, etc. What's really fascinating is that by just observing tiny little duplications anywhere in the genome, you can predict very accurately what Down syndrome does, at least at the IQ level. Of course, I'm not talking about specific disorders here, but at the IQ level, there seems to be a very general principle of the genome's effect on IQ. So this is a website that is still being updated. It's here, you can find it here, and I use it in the clinic almost daily, okay? So what you do is you just enter your CNV coordinates. I mean, okay, if it's a 22q, you don't have to enter it, you just call Anne, because she's got the answers. But in most cases, you pull up the CNV, you don't know what it does. And then you have like this report of 3 cases that's describing, well, what, 20% of autism, because that's what we have in the clinic. What is it? It's describing our clinical referral criteria. So you enter the start position and the stop position, and the model will give you the impact on IQ of that CNV if it's a deletion, and it's not yet running for duplication, but it's, as soon as we have a stable signal, we're going to have it running. What it also does is that it tells you if there's some DDD genes in there, and in the future we're also going to add those specific impacts, because those genes have an additional impact which is not 2.7 points of IQ per point of PLI, because a DDD gene is 30 points. It's a very large effect. Okay, so the last, do I still have some minutes? A few minutes. So let me just, okay. Just a little bit of a general concept of what this work has stirred up, at least in my lab, And so what we're obsessed with right now is this issue of, how do we really conceptualize the genome and psychiatric conditions? So, I was trained as a medical geneticist, and I did my residency in the mid-90s, at a time where we weren't really doing genetics anyway. I mean, there was we didn't even have genetic tests. We were, prescribing karyotypes and FISH analysis. But the huge success of genetics that we were taught really are summarized by this, medical genetic paradigm. You identify a gene associated with the disease. This is sickle cell disease, and you move your way, both directions. You move your way up the protein, the cell function, and then you understand the disease. And then also describing the disease helps you understand, the cellular, the interpretation of the cellular function and the protein. And, that medical genetics paradigm has been incredibly successful. It's been successful in cancer and all, cystic fibrosis. There's huge amounts of success stories. So it's a great paradigm. But I think that for psychiatry, what we feel is that genetic paradigm is irrelevant for psychiatry. So what I feel is that the individual function of genes, as we have been taught, so their cellular function, is irrelevant to psychiatry, because psychiatry may be a systems, it's a systems phenotype. Paranoia doesn't emerge from a cell. It probably emerges from a complex dysregulation of an entire system. And that's probably why we see these general models emerging where you delete any point of PLI anywhere in the genome, even if it's an immune gene or a regulator of I don't know what transcription factor. It's going to have the same impact on IQ, because what you're really doing is that you're just knocking out areas of a system, and the effect size of what and the general effect size is not related to the individual function of the gene. It's how you're distorting the system. So when you look at, for example, the autism genes that have been identified through the excess of de novo mutations present in autism cohorts, what you're really seeing, for example, are all these chromatin remodeling genes, and people are diving into these genes, but what do these genes do? They just regulate the rest of the genome. So these are epigenetic regulators, master switches. So you're back to a whole systems disease, and I think it really, I think we're going to have to shift completely the way we study the genome when we're looking at psychiatric disease, because even studying, synapses may tell you something about the condition, but maybe not. It's like saying, if I chop off the legs of every American citizen, the economy is going to go down. Okay, but what does it tell you really? What does it tell you on economy? Not much. It just tells you, yeah, if you have this massive structural alteration in every individual component of the system, then the system starts crashing. But it doesn't really tell you how the system works. And I think these chromatin remodeling, or even, for example, Fragile X, which is also a master regulator, are good hints at the fact that what we're really studying is a whole system. And if you look at this paper by Boyle and Pritchard that came out 2 years ago on the omnigenic effect of complex disease, what he shows is that, for example, if you look at schizophrenia, as the GWAS studies increase, every SNP is associated with schizophrenia. Every SNP will be associated with schizophrenia. So of course, most of them will have small effect sizes, but still, it really shows you that it's a systems disease. So yes, some of the mutations pop out because their effect size is stronger, but it's maybe not because they're closer to the function. It's just they have their hands on a broader regulatory function, and they're able alone to knock out or dysregulate the entire system. And I think this is what we're seeing for CNVs. As we're running more and more CGH, well, we're deleting the entire genome, and the entire genome is pathogenic. Okay? So in the very last part of the talk, how can you integrate this in understanding, for example, a condition such as autism and the CNVs in autism? So what we did is that we said, okay, CNVs are overrepresented in autism, but why are they overrepresented in autism? So we took the Simon Simplex collection and replicated what people already know. There's more deletions and duplications in the Simon Simplex collections. And then we ran a model selection procedure to say, okay, is there a model that can predict best how any CNV increases your risk for autism? And the best model is very close indeed to the model that I showed you for IQ. And there were other ones, but we just chose this one for simplicity. And what it tells you is that each time you delete a point of PLI, you increase your risk of autism by an odds ratio of 1.4. And for duplications, you do exactly the same thing. Each time you duplicate a point of PLI, you increase your risk for autism by 1.3. And you have this exactly the same whether you use siblings or unselected populations where you have IQ. And one of the big issues in the previous autism studies is that nobody had IQ in their control group. So in the Simon Simplex, there's no IQ in siblings. None of the family cohorts have phenotyped their siblings. And so this is the IQ distribution in autism, and this is the IQ distribution in our control population. And now that we have a very good handle on how duplications and deletions affect IQ as you delete or duplicate points of PLI, you can adjust for that. And what you see is that once you adjust for that, deletions, although deletions are highly overrepresented in autism, once you adjust for the effect on IQ, there's no more overrepresentation, or it's marginal. On the other hand, duplicating doesn't change. So as you duplicate, their impact on IQ is unrelated to the risk that they confer to autism. So I think that's the first really interesting point. And this has been replicated now in the missing data set, which almost has the same IQ distribution. So I'm just going to skip this. And if you look, so then you say, okay, deletions, you delete anything, you increase your risk for autism, but we think that we understand that it's mostly by decreasing IQ that you're driving this risk. Now, duplications, you're increasing the risk, but it's not IQ. So what is it? So you look at core symptoms. So we looked at the ADOS and the ADI. We looked at regression, which is not a core symptom, but there's people in our group that consider regression as a very important core symptom of autism here. And what does it tell you? Actually, haploinsufficiency decreases your risk of developing core symptoms of autism. So even though haploinsufficiency is widely overrepresented in autism, it's actually protecting you against core symptoms of autism. And I think that's a really good example of showing that you can have mutations in your patient for the opposite reason than what you think. So haploinsufficiency, you have less risk of regression, you have a better ADOS score, and it's significant even when you correct for IQ. And what haploinsufficiency does is it really impacts phenology. So phenology, each time you increase a point of PLI anywhere in the genome, your phenology drops. And so you have this general phenomenon of haploinsufficiency where you have cognition, haploinsufficiency, and then motor skills, which I'm not showing here, but motor skills, for some reason, it's a huge impact. And even if you correct for IQ, you still have motor skills that are impacted by haploinsufficiency. So I'm not saying that this is the reason for that's why every deletion is present in our autism population, but there's an underlying phenomenon that is driving the overrepresentation of haploinsufficiency, so deletions in autism. On the other hand, duplications, it's not IQ, it's not language, so duplications don't do anything to language, nothing. They don't really seem to do anything to core symptoms. The only thing they do is motor skills that we were able to measure. So why are they present in our autism kids? I have no idea. Is it because, you know, they walk late, so they're referred to the clinic, and if you're referred to the clinic, you need a diagnosis, and then if you have autism symptoms, you're going to get an autism diagnosis. Or maybe they do affect some autism core symptoms, but we need a general population to show it. But so basically, this is sort of the schematic of this, is that you have a kid who could meet autism criteria, but he's not referred to the clinic. He's doing okay. He's got autism core symptoms, you know, not too many CBCL, psychopathology problems. His IQ is pretty good, speech and motor, fine, not referred. You have the same kid, you throw a deletion on him, then all of a sudden his IQ drops, his speech drops, and his motor, and he's got motor delay. So that kid's going to be referred at 1.5 years of age because he's not walking, and then he's going to still be followed because he's going to not be speaking on time. And then, you know, people are around him, it's like, okay, what does this kid have? Oh, he's got autism. So what we think is that, you know, these deletions, although they don't, like haploinsufficiency doesn't affect core symptoms of autism, it gives an opportunity for the child to be referred for other reasons, and then he may get, it's increasing his probability of getting a diagnosis of autism. And for duplications, well, I don't know why they're referred to the clinic, but I think it's a cool question that we're going to dig into. And just to finish, and this is an open a call for collaboration. We have a new NIMH project with Boston and Philadelphia, where we're combining any existing data set, which is genotyped, with good continuous cognitive and behavioral measures, and then some genotype or whole genome data, to explore at a very large scale the impact of every, you know, rare loss of function mutation, and hopefully we can go to missense also mutations. So sort of get a genome-wide scan on the quantitative effects of all these mutations on specific cognitive and behavioral dimensions. Because when you start doing that, you realize that, you know, the mutations that you see in the clinic are doing funky stuff, not what you're expecting that they're doing, not necessarily what you expect. And so this is a 5-year project. It's just starting. So if you have cohorts like this, we'd be really happy for you to collaborate on this. And thanks for hanging in there. And these are the acknowledgements. Thank you.